Production of Antisera, that is, production of antibodies against specific antigens. This contains animal immunoglobulins or also called as immunoserum for human use in prepared from serum or plasma of immunized animals. Antisera are useful for the passive immunization purpose, and the purified product consists mainly of IgG type of antibodies. Animal immunoglobulins are used for therapeutic use in humans. These preparations in use include antitoxins, that means antibodies raised against specific toxins such as microbial and other animal toxins. For example, botulinum toxin, tetanus toxin, antisera against bacterial and viral agents, and antisera against the venoms of snakes, scorpions, and spiders. Remember that the antisera against the venoms of snakes, scorpions, and spiders is called as antivenins, whereas antibodies against different types of toxins is called as antitoxins. Animal immunoglobulins are administered intramuscularly, subcutaneously, or intravenously. Animal immunoglobulins are also used for the diagnostic purposes. The simplest example is its use in the detection of blood groups and diagnosis of diseases such as typhoid. Also, these immunoglobulins are useful in several serological tests. The method of production of antisera. These manufacturing steps are divided into several categories. The first step is preparation of the immunization antigen. Second is immunization of animals, collection of serum, adsorption of undesirable antibodies, purification, including the steps for removal or inactivation of viruses, formulation, and finally filling. There are certain animals which are used in the manufacturing of immunoglobulins. The animals used should be a species approved by the competent authority. It should be healthy and exclusively reserved for the production of immunoserum. These experimental animals include rabbits, horses, goats, and sheep. All these experimental animals should remain healthy. They should not show any symptom of the disease. Before the inoculation of the animals should not show the presence of any other type of antibodies that may cause the contamination of the final preparation. It should not show the presence of antibiotics also, and if there is presence of such antibiotics or other antibodies, then the animals are quarantined so that they should be recovered from the particular or there is a waiting period for the removal of antibodies or antibiotics. The production process. First step is immunization of animals. So for this, the antigen should be available in pure form along with adjuvant that may that may improve the antigenicity. Then the particular dose of injection is also decided and it is important, followed by first dose. The repeated doses, which are called as booster doses, should be given at a regular interval. So this protocol is standardized protocol. After immunization, after a particular period of time, the bleeding is done. Bleeding is the collection of blood or the collection of serum in a separate room. For this, the animals may be anesthetized. Blood collection is done through venipuncture or directly from heart, that is called as intracardiac puncture. The sterility of the product must be maintained. Nowadays, the animals are not anesthetized and the blood is collected by venipuncture, it will go to a machine that will automatically separate blood cells and serum. The blood cells are returned to the animal's body and the serum is separated. So this is possible through an automatic machine. Then the serum is collected in cold. Serum collected from different animals is then added to a bulk. It is called as pool testing. So collection of serum from different animals, and this serum is tested for the presence of any contaminant such as viruses. Then the next step is purification, which is done by ultrafiltration and possibly by ion exchange chromatography. The final product, it should be prepared according to the standards of pharmacopoeia that should be in line with dose, clarity standards and absence of contaminants. Then the product should pass through several quality control tests that includes measuring the antibody titer, its specificity, purity, pyrogen testing, protein content, and specific activity. Finally, the potency test is done in order to test the potential of the preparation. That means here the potential is measured in terms of its passive immunization, that is to neutralize specific type of antigen. The protective and therapeutic effect of the preparation is thus determined, in which the animals are challenged with specific toxin, and followed by this they are administered with antisera, and the potential of this antisera for the neutralization of toxin is then periodically observed depending on the development of symptom in animals. This step is also essential to decide the dose of antisera. This is the flow sheet for the production of antisera. The purified antigens which are required for immunization are such as venoms from spiders, venom from scorpions or jellyfish, venom from snakes, viral and bacterial antigens. In general, for venom from spiders, the rabbits are used. For scorpions and jellyfish venoms, sheep are used for immunization. Then horses are used for immunization by snake antigens, viral and bacterial antigens. After immunization, periodically there is antibody check from the serum of rabbit, sheep and horses. After getting the highest yield of antibodies in the serum, the next step is bleeding, which is also called as plasmapheresis. Followed by that, there is plasma pool, that means mixing of antisera obtained from different animals. Purification, that involves plasma fractionation, purification of immunoglobulin, then formulation, uh, in which there may be addition of several preservatives, then filling into vials or bottles, which is done aseptically. Followed by that, the quality control test that involves mainly potency test, then labeling, packaging and distribution.